My name is Jean-Phillippe Martin. I'm a senior software engineer at Verily Life Science. And today we'll be talking about things I worked on, our genomic pipeline with Apache Beam. So the idea for today's talk is I'll start by giving you the background, because it's about reading DNA, or sequencing DNA as we call it. I'll tell you about the little problem that we'll talk about today, the tools that we have to solve it. This is going to be, of course, Apache Beam. How we solved it, and then we'll spend a few slides showing how well it worked. All right, so let's get started. DNA. All right, we all have DNA. It's in every cell in our body. We get it from our parents. We have one from each parent. We write it as a long string of letters. It's 3 billion letters. They're going to be A, C, T, G. We have the technology to read people's DNA. I wouldn't say that we know everything about it. There's still a lot to learn. If you take two random people, you'll find that they differ in about 0.1% of their genome, of their DNA. So that's about 3 million letters. And DNA is particularly interesting because one of the reasons is that the cost has gone down a lot. So you may have seen this graph before if you've gone to any talk about sequencing DNA or reading DNA, but we'll go through it quickly. So the x-axis is time. The y-axis is cost. This is a logarithmic scale. The green line is the cost of reading the DNA, and as you can see, it has crashed. The comparison line in white is Moore's law. So the price has gone down even faster than Moore's law. And so now it costs roughly $1,000 to sequence somebody's DNA. So this is fantastic, and this means that we do it a lot more than before because now it's cheaper. Reading DNA is really nice for a number of reasons. It's not just about reading the genome and understanding the ancestry and figuring out the ways in which people are different. You could also sequence somebody's cancer tumor to try to find which medicine are going to work. You can also do other things than reading DNA using the exact same technology. So one of the things that people do is they read the RNA that is active in the cells as they're creating proteins. So what this does is that whereas the DNA is mostly static if you're healthy, so you get your DNA and then that's what it is, it determines the blueprints of your body. Reading the RNA tells you what this particular cell is doing at this particular time. So this is something that you can explore different tissues, different places in your body, they're going to be doing different things. And then at different times, it may be doing different things, perhaps healthy versus sick. And you do this using the exact same technology. So the price, the crashing of price means that we can now do this RNA reading also very cheaply. Another thing that people do is because this instrument works so well, we use it to measure other things. So we use DNA sequencing to find proteins. So the way this works, just briefly, is we can, and by we I mean the field, we can engineer hopefully I get this right. I'm a computer scientist, right? So we can engineer an antibody that will attach to this particular protein, and we can attach a bit of DNA to it. So what we do is we just mix this in the blood or whatever we're interested in, and then we sequence this, we wash the remaining liquid off, and then we sequence it. And so if that particular protein was there, then the antibody attached to it, and then we'll find the DNA that we're looking for. So you can read DNA in order to find proteins or things that are not directly related to people's DNA. Okay, so this is why I'm very excited about reading DNA. The point I wanted to make here also is that the cost of doing the lab part has gone down a lot. And in comparison, the cost of doing the computer part, the analysis part, has not gone down as much. So in relative terms, the computation is now more important. And so you want to spend a little bit of effort trying to perhaps make it go faster or cheaper, depending on what you're optimizing for. And this is amplified by the fact that we now have even more data than before because it's cheap, so we do it more. So computation is relatively more expensive, and there's also more of it. So we'll be talking about some computation. So now, let me tell you a little bit about how reading DNA works, just so I can motivate the problem that we're trying to solve. So a typical lab would receive perhaps 100,000 samples a year, and the sample would be perhaps a vial of blood. It could be other things. And the first thing you do is you try to extract the DNA from this and amplify it, get a higher concentration of DNA. Perhaps you'll have 10 times as much as you started from. And then we cut it into tiny fragments. And this is because the machines can only read short bits of DNA at all. So we need to cut it so the machine can access these parts. A lot of fragments, so perhaps 100 million fragments in this example. And so DNA, by the way, has a direction, so we don't worry too much about it flipping around. And then you put it in the machine. This is an example machine. It doesn't have to be this one. The machine is going to read these fragments, and there some things can go wrong. These machines are not perfect. So one of the things that can go wrong is duplicates. So bits of DNA can show up in the output as if they were there twice as much as they really are in the sample. And this is the problem that we're going to work on in this talk. So one of the reasons this can happen is there's an optical part in this process. So the picture here symbolizes a bit of DNA, and on the top are the reads. So you recognize these are the letters I told you comprise DNA. And if the thing is a little bit out of focus, it's possible that instead of seeing one dot of light, it thinks there are two dots. And so that would mean that each one of these now shows up twice in the output. And this can happen as much as for 25% of the reads. So it's significant. And it's a problem because when you do the analysis, you really care about the relative ratio of each particular bit of DNA. And so if these ratios are out of whack because of errors, your analysis may be wrong or slightly off target. So we want to detect these duplicates and remove them so that we can restore the independence assumption that underlies some of the analysis that you want to run downstream. Now, for the context, here is how the rest of the bits happen. So the first image here is in the machine. So remember, we cut the DNA into these small fragments. Then the machine is going to read from each end of the fragment going inwards. And it can only read roughly 100 bases down. And so they won't meet in the middle. They will just read a little bit, and then the machine has to stop. So you have these two reads that we know they're close to each other in the genome, and so we say that they're a pair. They go together. And of course, then we flip them so they're at the right spot. So the green box onward is what happens outside of the machine. This is now part of the computer stuff. And so the first thing we do is we flip them right side up, and then based on the letters that we see, we're going to, there's of course more letters than just shown on the slide. We're going to compare this to the reference genome and figure out where in the genome this particular read should be. So this is represented by the number here on the left, and there you see the dotted line. So the first one starts at position 100. Of course, we'd also say which chromosome it's at. Here, I'm simplifying for the talk. So you have the position for each. And then because we amplify the DNA at the first stage, you don't have just one strand of DNA in there. So it's possible that they overlap. And so you can use this overlap to perhaps find errors. So one of the errors that the machine can do, we already talked about duplication. Another error they can make is just misread one of the letters. So perhaps you have two reads like the two on the left here in the red box that overlap, and then you would expect the letters to be the same. And in this case, there is one place where they're different. So this could be because it's one from the father, one for the mother, or it could be because there was a reading error. And so if you have multiple reads that overlap, and you have removed the duplicates, then you can perhaps use majority rule or something like this to figure out what's the reality there and which part was an error. So this is an example where removing the duplicates matters. And yeah, so if I like this, maybe 50 gigabytes or so, just to give you an idea of the scale. So what this looks like in the computer, on the top I have the symbolic representation. You have the helix of the DNA, and you have the reads that we found. So on the computer, from our point of view here today, it's just a record with these 4 fields. And importantly, if two reads come from the same fragment of DNA, so they're both read from at different ends of the same fragment of DNA, we say that they're paired, and we're going to give them the same name. This is how we know they go together. And there is a paired flag that we're going to set to yes so that we remember that these go together. I represent them with warmer colors for the reads that are paired and a cooler color for the reads that are not, but you don't need to remember this. So All right, so here's the takeaway from the biology. So you don't need to remember the biology, but all that really matters from the point of view of the computation we'll be talking about is that the data structure we work with is a set of read records, and these read records have the fields that are here on the screen. Importantly, they have a name, a pair flag, and a position. And if two reads have the same name, that means that they are paired, and their pair flag is going to be set to yes. The task is to set the flag that's circled here called isDuplicate, because we're looking for the duplicates. I will tell you in the next slide what are the rules for doing this. And the output is of the same form. It's a set of records, but now we filled in this form, this field. All right, so the rules. Now this slide looks a little bit busy, but bear with me, this is actually fairly simple. We're comparing reads, and then they can be paired or not. So there's three cases. Either they're both unpaired, or both paired, or one of each. We'll do each case at a time. They're just the different rows. So the first case is unpaired. So suppose you have two unpaired reads. The rule that we use is simply if they both have the same position, then we say they're a duplicate, and we're going to mark one of them. And I represent this in the example in the middle column. You can see the green and blue reads here. I align them to the same horizontal position. I'm using this to symbolically represent the fact that they have the same position in the genome, so their position field has the same value. So they're duplicates, and so I circle one that we're going to mark as duplicate. The column on the right shows an example of a non-duplicate. They're offset. They don't have the same position, so we don't mark them. Now for pairs, the middle row, the rule is that both positions must match. So if you have paired reads, if you have two pairs, then the position of each element of the pair must match with the corresponding element from the other pair. So you can see in the example here, I have a green pair and an orange pair, and they must align on the left and on the right. On the counter example on the right, you can see if any one of them is offset, then we don't mark anything. The final case is mix. If you have one paired read and one unpaired read, then it's only the unpaired read that's going to get marked, and it's going to be marked if its position matches either of the elements of the pair. So it's always kind of the same rule. It's just if there's only one position, it's the position must match. If you have two positions, both positions must match. And if it's a mix, one of the positions must match. So that's the rule that we have to implement. And now, before we start talking about tool, I want to give you just a quick kind of overview of the landscape of things that you could use. This is very high level. It's kind of my own way of thinking of it. So the way I think of it is on the one hand, on the left, you have what I'm calling servers as a service. This is where you use the cloud just to rent a computer and run your thing on it. And so you can take an existing program and just put it there and it'll work just fine. This is the easiest thing to do because it's very familiar. And so this is great. For example, if you already have some existing legacy already written software, you can just run it there as is. You would perhaps use a tool like Kubernetes. In life science, we have developed our own tools. There is Cromwell, and at Verily we put together a dsub, which is it's for our users. It's a simple tool for running things on the cloud, on the server. Or you may want to go a little bit further down the abstraction route and use what I'm calling a database-centric approach, where you use a database abstraction that the cloud can give you, such as BigQuery. And so here, this is great when your data is mostly static. Perhaps you've already done the analysis, now you just want to present in a fancy way, and you want to be able to have multiple computers serving the data, so lots of people can go and look at your results. And the one on the right is pushing the abstraction a little bit further. Now, that's the Dataflow languages. So the Dataflow languages allow you to get a lot of parallelism, and the idea here is that we are now representing your computation. This is for your computation. We're representing it as a graph where the nodes represent things that happen to the data, so computation, and the edges represent the data flowing. That's why we call them a data flow language. So in this example picture here, we have three different things that happen to the data, and the data kind of flows from one to the other. And there are multiple such languages. There's Apache Beam that we'll talk about, and you may have heard of Spark also. So you get parallelism because each of these 3 steps here in the picture can be done at the same time. Each of these are maybe different computers running at the same time. And also each individual step can be done in cooperative manner by multiple computers. So let's give you the punchline of the talk already. So we started from a non-optimized single server running this thing, and then we wanted to write it with Beam, and we did, and the result is 5 times faster at only twice the cost. So we're pretty happy with the result. But let's see how we did this. Why did we choose Apache Beam? Apache Beam is good when you're working on a large set of elements that are all of the same type. This is the case here with the genomics read. We have a large number of them. We can process them in parallel. This is a common pattern in life science, and you may recognize the same pattern in your own field. It's not uncommon to have many receipts or images or purchases or clicks or views or ad impressions, things like that. The best way I know to explain Beam is through an example, so we'll just jump right in. So suppose what you're trying to do is, still in the genomic context that we're talking about, suppose we want to know for each position in the genome, how many reads do we have at that particular position? So in the example here, we go from left to right. On the left, we have storage with our set of reads. There are 2 blue reads at position 12 and a red read at position 20. So the way you do this with Beam is you would start by grouping by position. So group by would put elements together that match the thing that you specify. So position in this case, so you end up with 2 groups, a group at position 12, a group at position 20. And the next step you would do is you would say, now I want to count how many elements are in each group. And so you get your answer. At position 12, I have 2 reads. At position 20, I have 1. We're done. That's what it looks like. It's a very simple example, of course. But at least I didn't give you a word count. Now, this is the abstraction, right? So what happens when you run this in Beam is that your code remains simple. So your code is, I put pseudocode here on the left. It matches kind of these simple concepts. But the runtime can actually leverage the simple description to give you parallelism. So you saw whereas before we had 1 yellow dot representing 1 computation, when you hit run, it ends up being multiple machines. I put 2 on the slide for space. So what can happen is, the compiler recognizes that you're trying to do an operation on every element. This can be done in parallel, so let's do it. It recognizes, you're reading these files from the cloud. Well, it so happens that the cloud replicates files for availability, so we can actually read from different replicas to get more performance. And it will do all of these things for you transparently. One cool thing it can do also is actually it can watch your computation as it goes and potentially add or remove computers to tune things as it's going. So I wanted to say, in case you're familiar with MapReduce, this may look very familiar to you. And so indeed, Apache Beam is a generalization of MapReduce. So in the sample example, it looks the same, but Beam differs in, I would say, 2 ways. The first is that instead of just being 1 map and 1 reduce, you can do an arbitrary number of these together in the same program without having to do anything special. So it doesn't have to be 2 steps. And the other difference is it has more operations than just these 2. So let's go through some of the operations that Beam can do. So here's an example Beam program. It's a graph. They all look like graphs. This program is completely artificial. It's just there to show the different things that Beam can do. So the first computation here marked A, I wanted to show that Beam can have multiple inputs coming into the same computation and multiple outputs. So you're not limited to simple graphs. And when you send to multiple places, it could be you send the same data to everybody. You have different sub-computations that you need to do in the same input. Or perhaps you send different subsets of your data to different sub-computations. Perhaps, I don't know, all the green images get some processing, and all the blue images processed in a different way. So that's multiple inputs and outputs. The next dot, the B circle here, shows the map operation. You apply an arbitrary function to each element. So in the picture here, I'm just bolding them. Of course, you can write arbitrary code. You just write whatever code you like to process the elements, and when you put this in Beam, you will know to put it on multiple computers and apply this to all the elements in your collection. You can also add or remove elements from a collection. So this is the next dot here. You see I've added the Pac-Man shape and removed another. The next one, D, is combining. So combining, here I'm showing just the count. So we have 3 shapes, and I get the count. There's a number of built-in combiners. You can do a max, you can do a sum, minimum, obviously. But the neat thing is actually you can write your own. So any piece of code, so long as it's associative and commutative, the Beam will know how to run it efficiently on multiple machines and run a little computation tree, aggregation tree, and get you the right answer efficiently. And you don't need to do anything. You just specify the aggregation function. The final one on this slide, there's actually more, but the final one here we're talking about is group by. You've seen group by before, where we're grouping by a position. So it takes a set of elements and it gives you a set of groups of elements. Group by in Beam actually has another really nice feature that I just want to mention now, and even though we're not using here, is that Apache Beam can run in a streaming mode. So instead, whereas in a normal, usual computation, you have some input and you run it on your program and you get an output and you're done. In a streaming computation, the program just keeps on running forever, conceptually, and you have inputs coming in as streaming in. Perhaps they're publish, subscribes, events, or clicks, or things happen in the world, and then your program computes things and they spit out the back continually. So you can use group by for this. You can group by time windows. So you can get like a daily summary or something, or you can also group by and get a sliding window. So, you know, the number of sales in the past 10 minutes or whatever, things like this. So, okay, so this is the overview of what Beam can do. There is more, but hopefully this gives you a good taste and a good idea of what it can do. And when you write Beam, you can write it in actually multiple languages. You can have your choice. There's Java, Scala, Python, or Go. And I used Python, and here's what the sample program looks like in Python. It's fairly straightforward. The one thing to notice is that the group by is now 2 lines. What happens is group by doesn't take arguments. Instead, what you do is you write your data as a tuple, where the first element of the tuple is what you want to group by. And then you just call group by, or group by key, and it's going to group by the first element. So you write these 2 lines to do your group by. And you can see here the file names start with GS. That's a path for storage on Google Cloud Storage. So I could say a lot more about Beam. I just want to focus on my favorite features. And so the first one here is that Beam is a little bit unique in the sense that when you write a Beam program, it will actually kind of cross-compile it to different runners, to different other distributed systems. So the one on the left is not labeled, but this is Google Cloud Dataflow. So you would use this to run it on the cloud. You can also run it on Apache Flink or Spark or a number of other ones. So this is great because this means that you write the same program, and you could run it on your local cluster, say using Spark, or you could run it on a Google cluster using Google Cloud Dataflow, or you could run it on somebody else's cluster using one of these other systems. And if one of these systems develops some great innovation and becomes super fast, you can just choose to use this one. So that's really nice. The other thing is the auto-scaling I already hinted at before. So this graph shows this kind of in a graphical way. This is what you actually see when you're running. Here it was just a toy program. But the idea is that it will add and remove computers as needed. So there are 2 lines here, so you may be curious. The green line represents the number of computers that the system decided was optimal. And the blue line is the number of computers that were actually running at the time. So you can see the blue line just kind of quickly matches up to the green line. It just takes a little bit of time to start and stop the computers. And you can see this particular computation, it has multiple phases. There is some sort of perhaps setup at the beginning where you have a small number of computers. And then the computation ramps up and the system adds machines as needed. And it looks like there's a third phase at the end where we're doing something else that doesn't need quite as many machines, and so they quickly cut down the number of computers. And this is done automatically. All you have to do is enable auto-scaling, and then you can just see what happens. The third thing I wanted to highlight about Apache Beam is the many connection options. So I already mentioned that you can read from a file on the Google Cloud with Google Cloud Storage. You can also read from other people's clouds. You can read from a database. You can read events from a publish subscribe system. You can even, this would be for the streaming case I mentioned before, you can even connect it to another Dataflow language that's going to be streaming events at you. So you can imagine running Apache Kafka to get some events streaming through the system, and you perhaps for some reason already wrote this in Kafka, then you want to do some additional processing in Apache Beam. Well, you can connect the two and the events flow through, and now you can do your streaming computation. And when you're running Beam, if you're running with Dataflow, then this is what it looks like. I know that the picture is perhaps too small to see the details, but I just wanted to give you an overview anyways. So the biggest part here on the left is a graphical representation of your program. That's the one you gave in when you type in your program. Beam programs are graphs. And each box is labeled with the name that you gave it. These are your functions. And it gives you some information about whether it's running or not and how fast and so on. And on the right, you have some overall statistics about your job. It tells you how many computers are running it, so that's the worker count. It tells you how many resources have been consumed so far. Some options that you specified on the command line are repeated here. And then you have the auto-scaling graph in the middle that I already explained before. So as your computation is running, you can see how many computers are assigned to it. And you can choose to use auto-scaling or not. If you don't, it's just going to be a straight line. So that's our quick introduction to Apache Beam. And hopefully I made you want to see more and learn more. And now we'll go through this example of marking the duplicates using Apache Beam so you can see what it looks like in action. So just a quick reminder, right? We have this set of read records, and we want to update the isDuplicate fields. And the rule is that if they're unpaired and in the same position, we mark them as duplicate. If they're paired and both at the same position, and if they're a mix, then if any of the position matches, we mark it as duplicate. That's the rules. Let's go. So here's the first Beam program to do this. So our input on the left is the set of read records, and the first thing we're going to do is check whether they're paired or not. We have the paired flag, then we're going to send it to the different branch. So let's start with the blue branch at the bottom, where the case when they're not paired. So when the reads are not paired, we're going to use group by to put together all the reads that start at the same position. So we're grouping by position. And then we look at each group, and we look at how many elements they are. If there is more than one, we found duplicates, so we mark them. And then we're done, goes to the storage on the end. So our output, set of read records again. In the case where they're paired, we need to do a little bit more work. The first thing we do is we group them by name to put the pairs back together. Remember, if two reads are paired, they have the same name, so doing this, we get the pairs back. Then once we have the pairs, we group the pairs by their position tuples, so the position of each half. And if you have more than one in the same group, that means that both of their positions match, so they're duplicates, so we mark them, and we're done. So this is hopefully fairly reasonable. I'll go through an example to make sure that we're really on the same page. We'll start with the unpaired read example. So here we have three reads. There is blue, green, and dark blue. And the blue and dark blue are both at position 100, and the green one is at a different position. So we checked, yes, they are not paired, so we go in the blue path. And the first thing we do is we group them by position. So we end up with these two groups here. There's a group of position 100 and a group of position 400. You can see that the first group has more than one element. These reads are duplicates, so we are marking every one except the first one, representing this by circling them in the picture. This means we set the field to true. And we're done. Now, in the case of paired reads, this time here I give you 4 reads, 2 yellow and 2 red. The 2 yellow have the same name, they're paired, the 2 red likewise, and their positions are 100 and 400. So the first thing we do is we group them by name. So you can see now we put the pairs back together. So you have the yellow pair and the red pair. If there weren't any more, they would be there. The next thing we do is now we group by position. And so it so happens in this example that both pairs have the same positions. They're at 100, 400. So this group has more than one element. It means we found a duplicate. We mark it. We're done. So this is great. And it would work, except that there's a case we didn't go through. You may have noticed this. You may have been wondering while I was presenting, what about if we have reads that are paired mixed with reads that are unpaired? There's a rule there that says we have to mark some of them. So we need to add this for the program to work. So let's fix the program. So here's the exact same program, except now I removed the details from the boxes and I just put a one-line description of what each box does. But it's the same program. So the fix is the following. You first check if the reads are paired or not, and then you send all the reads down the unpaired check, because when we have the unpaired reads, we need to mark them as duplicates if they match another unpaired read, but also if they match a paired read. So we're kind of merging the red and the blue paths down here at the bottom. That's why I put it in purple. So now we can check the unpaired read correctly and write them down. And the top part remains, of course. So the paired reads, we still need to put the pairs together and do the processing and everything. So this is a correct fix. This will work. I've run this. But now you may be wondering, well, these paired reads are going through both paths. Looks like we're doing some redundant computation. Could we optimize this a little bit? Could we change the program such that instead of sending all the paired reads to check, we perhaps eliminate some paired reads that can't possibly be duplicates in this first purple step, and then only send the remainder to the red step instead of sending them all? And of course, the reason I'm suggesting this is that, yes, you can do it. The key insight here is, what can we do when we only have half of the pair? So if they're unpaired, of course, we can do the whole thing. In the example here, right, I have the green and the blue read. I'm aligning them vertically to represent the fact that they have the same position. These are duplicates. If you have paired reads, here, for example, the yellow and orange, I put the dotted line to show that this is half of a paired read. We can see that these two are the same position, they're aligned, but we don't know what happens to the other half of the pair. So we actually don't know whether they're duplicates or not. They could be duplicates, or they could be non-duplicates. At this point, we really can't tell. The orange read at the bottom instead, this one, we know it can't possibly be a duplicate, because remember, the rule is that both halves have to match, right? And at this position, there is only one read. So we know that because this half doesn't match, it doesn't matter what happens to the other half, this particular read cannot be a duplicate. So we can eliminate it. So that's the insight. We only need to take the reads that are in this middle case and send them for further processing to double check if they're paired or, if they're duplicates or not. The rest, we're done. And so just quickly, here is what the program looks like. I know the slide looks busy, but this is very similar to the program we've seen before, worked through together before. It's just vertical because I had to fit it. We start at the top left, the reads come in, we group them by position, and once we have this, we can correctly mark all the unpaired reads that are duplicates. And then what about the paired reads? Well, we look, is there more than one at this particular location? If there is more than one, then we go yes, and the paired reads go to the red path on the right. So because these are the ambiguous case. There is more than one, they could be duplicate, we don't know. If there is one or fewer, then everything goes to the output. So we're done with those. And the red path is the same as before. You put the pairs together, you check their positions. If there's more than one of the same position, you mark them as duplicates. We've seen this, it works. All right, so just to make things a little bit concrete, here's what it looks like in Python. This is actual code taken straight from my editor. It's incomplete, of course. The full program wouldn't fit. But this is just to give you a feel. So the text that's bolded is actually comments. It's the name of the stages of the boxes. And so this is what the names that will show up in the GUI. And then I call the code. Yes, they're all in the Beam namespace, and you can see what we do. So here there's a function I'm calling keep only if has mapped mate. So I only keep the paired reads, and then the step one will put a key in it that represents the name, and so I'm grouping by name, and then I'm putting a key that represents the positions, and then I'm grouping by position. This is step 4. This is exactly as we described in the box. Then we mark the duplicates, and we're done. So, of course, we need to fill in the functions. There are plenty of examples on the web, but this is what this particular program looks like, and it gives you a flavor. So there's one more thing I did that I need to mention for completeness. By default, when you run this on the cloud, this is for Dataflow in particular, it will assign each worker 250 gigabytes disk. Turns out for this work, this was more than necessary, so I shrunk it to 85 disk. So now we need less storage, and so it's going to be cheaper. And here are the results of the optimization steps. So what I'm comparing here is the original fixed program that we've seen in the talk that's still running Beam, and then the optimized Beam program. So this is Apache Beam on both sides. This is just the impact of these two optimizations we did. First, shrinking the disk size, and second, the algorithmic optimization I walked you through. And so what we can see is that we're 43% cheaper, so almost half the costs with these small changes. A third of that comes from shrinking the disk, and another 2/3 from the optimization that we walked through. And the other thing that I wanted to do is compare this program against our original program that was running on a single server. So the single server program was running off-the-shelf software using genomic libraries and stuff, and I must emphasize it wasn't particularly tuned for performance. And I'm comparing it to our optimized Apache Beam program. And of course, this is our own versions, right? I'm not making any promise that the same thing will happen to you if you port your programs to Beam. This is just what happened to us. And what happened to us is we're 5 times faster, 5.6 times faster. The cost also went up, but it went up only 2x. So I have this graphically here on the slide. The green bars show the time, server on the left, Apache Beam on the right. So you can see the time just crashed down. And the cost on the right in blue, so again, server is left, the Beam version is on the right, the cost roughly doubled. But the absolute numbers are not that high. So what's not in these, we like to focus on the numbers, right? But another thing that's really important, and perhaps even more important, is that the program now is a lot simpler. Because this program now just needs to focus on the business logic. In our case, that is marking the duplicates. All the parallelization and the distribution and the fault tolerance and all that stuff is done by Beam and Dataflow. We don't have to worry about it. So this is something that I really appreciate. And then we have this other side bonus, which is we have now a trade-off between speed and cost, because we can choose to set the number of workers, the number of computers that are running our program. So these numbers I gave you are for 90 workers, but you can also run it for different numbers of workers. So what I plotted here is, so each dot represents the average of 5 experiments with a set number of computers. So I went from 15 to 120 workers, and I measured for each of these, I measured how much it costs and how long it took. So the x-axis is the cost, that's kind of how much you put in, and the y-axis is the runtime, so wall clock, so just measured overall. So you want to be kind of left and low. And what you can see, and this is actually a very common pattern, is that initially as you add workers, your performance improves dramatically and your cost only increases a little bit. And that's because there are parts of your program that parallelize really well, and so as you double the number of workers, these parts take only half the time, and so now your program's a lot faster. In an extreme case, you could imagine that perhaps all of your computation is like that. And if that were the case, then when you double the number of machines, your running time would completely halve, and because you're billed by the second, your cost would be the same. So you'd be putting twice as many computers, be going twice as fast for the same price. So this is the ideal case. Here, of course, in reality, there's a little bit of a slope. And what you see is, as you push this further, as you put more and more workers, now the parts that parallelize well are already shrunk to a small amount of time, so they don't represent a large part of the computation time. And the remaining parts perhaps don't parallelize as well, and so they're going to be relatively larger part of the time. And so the curve flattens up. So that's what you see. It starts out really steep, and then it flattens. And at which point is best for you really depends on how much you value time versus money. This is a trade-off that only you can make. Another way of representing the exact same data is as a ratio. So you can look at how much more expensive than the thing we started from and how much faster. So here I put in red the original program I started with. The blue dots are the same as before, so every dot represents the average of 5 experiments for a given number of workers. And so the dotted line represents kind of what you might expect if you were taking the original program and just if you were able to run it on 2 machines and get it twice as fast. So the dotted line represents this linear speed up of, if you double the cost, perhaps you'll get twice the speed. But of course, in our case, this is not the same program. This is a completely new program that is written, cloud native in Beam, and has different characteristics. And so what we see is that it shifts kind of up and to the right. It's more expensive, but much faster. And as we add to the number of workers, it goes up. If I were to add more, I expect this would flatten out. I just didn't run the experiments with more. So that's what it looks like. Just a few takeaways. So if you can remember just one thing about Apache Beam is think of it as it's an abstraction about how the data flows through your computation. And putting your computation in these terms allows it to get really high degrees of parallelism and performance. This is really good when you're computing with lots of data items that are the same type. This is a common pattern in life science. You may also see it in your field. The most common operations are group by and map or parallel do. And so group by will put together elements that match in some way that you specify. And the other one is when you want to do an operation on every element in a group or in the collection. There's more, as you've seen. Apache Beam is great when you're writing a new program. You can write your program in this cloud-native way. If instead you already have a large amount of software that you can't modify, you want to run as is on the cloud, perhaps you want to use something else that lets you just rent a computer and run things there, like Kubernetes or our very own dsub, that's an open source tool.